Usually, when we set out to build a map, we expect the landscape to just kind of, sit still for us.
Yeah.
You send a satellite into orbit, you take a photograph of a mountain range, and that mountain is going to be in the exact same spot tomorrow.
Right, yeah. It's static. The physical world mostly cooperates with cartographers.
Exactly. You take another photo, you stitch them together, and boom, you have a map. But then you zoom in. You step into the microscopic world of biology, specifically inside a single living cell, and suddenly that mountain is moving.
Oh, it's constantly moving.
Right. It's changing shape, it's dividing, it's looping back on itself. And to make matters worse, the cameras we use to take pictures of this moving landscape are, well, they're all slightly distorted.
Which is exactly why we're looking at a completely different kind of cartography today. We are looking at a breakthrough called CONCORD, detailed in a recent Nature Biotechnology paper by Qian Zhu, Zevjay Gartner, and their team.
Okay, let's unpack this, because today's deep dive isn't just about biology. It is a masterclass in how curating information actually beats just, throwing massive computing power at a problem.
Yeah, it solves one of the most frustrating data corruption problems in modern science. And it does it not by building a massive supercomputer, but by completely changing the diet of the AI itself.
I love how this flips the current tech narrative on its head. But before we get into how CONCORD changes the AI's diet, we really need to understand what it's trying to digest. You mentioned earlier that cells form a geometric landscape.
I do, yeah.
Let's make that concrete for the listener.
Yeah.
When we say a cell has a state that forms a shape, what are we actually visualizing here?
Think of a cell's DNA like a massive control board. You've got roughly 20,000 switches. These are the genes. At any given second, a specific combination of those switches is flipped on and the rest are off. That specific pattern of on and off, that is the cell's state. Now, if you map millions of cells based on which switches are on, they don't just form a random cloud of data. They form distinct physical geometries.
So we're basically taking a spreadsheet with 20,000 columns and turning it into a 3D topographic map.
Precisely. And the shapes on that map tell a story. Like, if an organism is in homeostasis...
Meaning it's just maintaining its normal, stable, everyday life.
Exactly. In homeostasis, its cells group together into tightly packed, discrete clusters. You'll see a distinct cluster for muscle cells over here, and a completely separate cluster for nerve cells over there.
But biology isn't always stable. Like, what happens during development?
The geometry completely changes. If you look at a developing embryo, the cells start as generic stem cells and then physically branch out in the data. They form structures that look exactly like trees.
Wow, literally like a branching tree.
Yeah, a single trunk splits into branches of intermediate cell states until they reach the tips of the branches, their final terminal identities. And if a cell is preparing to divide, regulating its cell cycle, the data actually forms a continuous loop. It circles back on itself.
So you have clusters, trees, and loops. And the tool scientists use to read those 20,000 switches is called single-cell RNA sequencing, or cRNA-seq. Basically, they crack open a single cell, capture the RNA, which is like the active transcript of those flipped-on genes, and sequence it.
Right. But here's the major roadblock. If we want to build a master map of all these clusters, trees, and loops across the whole human body, we can't just run one giant experiment in one afternoon.
Yeah, we have to combine data from hundreds of different labs all over the world.
Exactly. And this is where the cartography kind of falls apart.
Right. So imagine trying to build a panoramic 3D map of a city, but you aren't allowed to take the photos yourself. You have to rely on thousands of photos submitted by different tourists.
That sounds like a nightmare.
It is. Some were taken on a cloudy Tuesday in December with a vintage film camera. Others were taken on a bright summer noon with a brand new smartphone. If you just mash all those photos together, the city looks distorted.
Yeah, a building might look blue in one photo and gray in another.
Exactly. Not because the building changed, but because the camera lens changed.
In computational biology, that different camera lens problem is known as the batch effect. It is technical variation. So maybe one lab used a slightly different chemical reagent, or, their sequencing machine was running a little hotter that day.
And it completely obscures the true biological signal.
Completely.
So just to do a quick mini recap here for you listening, we have these beautiful hidden geometries of life clusters, trees, loops, but our view is fogged up by lab noise. The camera lenses are distorting the buildings. Now, scientists have tried to fix this before, right, with older tools?
They have. Tools like PCA, Harmony, or Surrot. But they rely on very rigid mathematical assumptions. PCA, for example, tries to squash the data down by finding straight lines of variance.
Which brutally flattens the nuanced curves of those biological trees.
Right. Harmony tries to mathematically force datasets to align, but it often overcorrects. It blurs the map so much that you lose the subtle, rare cell states.
And Seurat?
Conversely, Seurat sometimes undercorrects, leaving you with fragmented islands. The map ends up clustering cells based on whether they were sequenced in Boston or Tokyo, rather than whether they are a heart cell or a lung cell.
Okay, so the brute force math wasn't working, which logically led scientists to try AI, specifically a very buzzy technique called contrastive learning.
It's a brilliant concept in theory. Contrastive learning doesn't require humans to manually label millions of cells. It learns by playing a matching game within small batches of data, which we call mini-batches.
How does that game actually work?
So the AI takes a cell, digitally masks part of its data to create a slightly altered version, and says to itself, okay, these two are the same underlying thing. They're positives. Then it looks at the other cells in the mini-batch and says, these are negatives. They are different.
Okay, I follow.
By constantly comparing positives to negatives, it figures out the robust underlying rules of what makes a cell unique, totally ignoring the superficial noise.
I have to push back here, though. If the AI is essentially playing an ultra-fast game of spot the difference, why didn't it immediately solve our map problem? It sounds like the perfect tool to look past the camera lens.
Because of how we were feeding it. The learning mechanism is sound, but traditional AI uses something called uniform random sampling. It reaches into the massive pile of global data and just grabs a random handful of cells to put into the mini-batch room.
And let me guess, grabbing randomly creates total chaos.
It creates two catastrophic failures. First, it causes a complete loss of resolution. If the random sampler grabs a specialized immune T cell and a generic skin cell, those two are so fundamentally, wildly different that the AI doesn't have to work hard to tell them apart.
It's like asking you to spot the difference between a bicycle and a 60-story skyscraper. You take one glance, learn the broadest possible features, and you're done.
Ah, exactly. The AI gets lazy. It learns the big picture, but becomes completely blind to the subtle intermediate cell states. And those intermediate states are the actual branches of the tree that tell us how a disease develops.
Wow. Okay, and what's the second failure?
The second failure is even worse. By randomly mixing cells from completely different data sets, different labs, different camera lenses, into the same mini-batch, the AI accidentally learns the batch effect itself.
Wait, how? Doesn't the contrastive learning filter that out?
No, because it thinks the noise is the signal.
Oh, no.
Yeah. If the AI sees a cell from dataset A and a cell from dataset B, and the most mathematically obvious difference between them is the technical static from dataset A's specific sequencing machine, the AI says, aha, this static is what makes this cell biologically unique.
It literally encodes the camera lens instead of the building.
Exactly.
Okay, let's lock this in with another quick recap for the listener. Contrastive learning is a sports car, but uniform sampling is basically putting sugar in the gas tank. It forces the AI to prioritize obvious, lazy differences, and it accidentally memorizes the lab noise. Which brings us to the actual breakthrough. How does CONCORD fix the engine?
Well, by completely ignoring the engine and redesigning the fuel pump, the researchers didn't build a bigger, more complex AI. They completely rewrote the rules for how cells are sampled. They created a joint probabilistic sampler with two massive innovations.
Let's hear the first one.
The first is called dataset-aware sampling.
Okay, I have an analogy for this. Let's imagine the AI's mini-batch room is a VIP nightclub, and it's trying to learn how to identify the subtle features of people's faces. The CONCORD algorithm acts as the bouncer at the door.
I like where this is going.
To solve the batch effect problem, the camera lens problem, this bouncer looks at IDs. On any given night, he only lets in people from the exact same town. If you're from the Boston dataset, only Boston cells get into the club tonight.
That is spot on. By restricting the mini-batch to a single dataset, the AI inside the club is physically prevented from comparing a Boston cell to a Tokyo cell. It literally cannot learn the batch effect because it never sees two different batches side by side.
But hold on, if the AI spends all night only looking at people from Boston, doesn't it just memorize the Boston accent?
Yeah.
Doesn't it just become incredibly biased toward that one single data set?
That would happen if it stayed on Boston forever. But CONCORD Chef.
Oh, it shuffles the batches.
Right. After training on a mini-batch from Boston, it instantly pivots to a mini-batch entirely from Tokyo, then one from London. If the AI started to encode a specific technical bias from Boston, that bias is immediately disrupted and mathematically overwritten the moment it processes Tokyo.
That's brilliant.
Yeah, the only patterns that survive this constant rapid-fire shuffling are the genuine underlying biological truths, the core gene pathways that exist regardless of what city you're in.
That is incredibly elegant. But that only solves the noise problem. What about the laziness problem? How do we stop the AI from just comparing the bicycle to the skyscraper?
That is the bouncer's second new rule, hard negative sampling. The bouncer doesn't just let in random people from Boston. He specifically looks for identical twins, or people who look incredibly similar, and forces them into the club together.
So now the AI is staring at a room full of closely related cells from the exact same data set.
And suddenly the lazy route is totally gone. The AI can't use their hometown to tell them apart because they're all from the same place. And it can't use broad, sweeping differences because they look almost identical.
It's forced to look at the tiniest detail.
Exactly. A fractional change in the expression of a single gene just to tell them apart.
To dig into the mechanics of that for a second,
Yeah.
how does the algorithm actually find these twins in a data set of 1 million cells? I mean, it sounds computationally exhausting to measure every single cell against every other cell before you even start the AI.
It is, which is why CONCORD uses 2 different modes to do it efficiently. The first mode is called KNN, or K-nearest neighbors.
How did that work?
Imagine a massive social network graph. KNN builds a localized web, finding the specific cells that are physically closest to each other in the high-dimensional space. It pulls them into the mini-batch, alongside just a few random distant cells to maintain a sense of the global map.
But even building that localized web takes memory. You mentioned a second mode that bypasses the web entirely.
Yes, the HCL mode. It relies on a mathematical technique called Monte Carlo importance sampling.
Let's translate that. Monte Carlo sounds like a casino.
It's actually a great comparison. Instead of manually mapping the entire population of the nightclub to find the twins, which takes forever, Monte Carlo sampling uses probability.
It guesses.
Sort of. It mathematically estimates which cells are likely to be hard negatives based on their distributions without explicitly calculating the distance between every single pair. It approximates the required learning difficulty, the expected loss, directly in the math.
So it's like guessing who the twins are by rapidly sampling small crowd sections, which saves a massive amount of computer memory.
Exactly. And all of this brilliant sampling leads to the paper's ultimate mic drop moment.
Oh, lay it on me.
Because the bouncer is doing such an impeccable job of curating the room, the AI inside the club doesn't need to be a genius. CONCORD achieves state-of-the-art biological mapping using a neural network with just a single hidden layer.
Just one. That's almost unheard of today.
It really is. For context, a hidden layer is where the AI does its actual processing, transforming the raw input data into the final map, which we call the latent space. Most modern AI models stack dozens, sometimes hundreds of these hidden layers to brute force complex problems.
Right, we're in the era of ChatGPT where models have billions of parameters and require server farms the size of football fields just to run. And CONCORD maps the fundamental geometry of life with one hidden layer.
It proves that if your data diet is pure, the brain digesting it can be incredibly simple. And because it's so simple, the final map, that latent space, is clean, robust, and mathematically interpretable by human scientists. It's not just a black box.
Okay, let's take a breath and connect the dots again for everyone listening. We started with a moving landscape obscured by the fog of different lab cameras. Older methods tried to map the fog away and broke the map. Contrastive AI tried to learn the map but got lazy and memorized the fog.
Yeah.
And CONCORD fixes this by changing the data diet. It uses dataset-aware sampling to isolate the fog and hard negative sampling, finding the twins, to force a remarkably simple AI to learn the deep, subtle truths of biology. So ask yourself, as you're listening, do you learn better by comparing two entirely different things or two things that are almost identical? But the theory is just theory. What happens when this simple AI hits the genuinely messy reality of living tissue?
Let's start with a foundational test case. The C. elegans worm. In biology, this microscopic worm is legendary because its development is almost entirely invariant. We know exactly what its cellular family tree should look like, from a single egg all the way to a fully formed adult.
Okay, so it's the perfect test subject.
Right. But when older dimensional reduction tools tried to map early-stage C. elegans embryogenesis using single-cell data, they failed.
How did they fail? Did they scramble the tree?
They left disconnected gaps in the early stage trajectories. Because the transition between states is so incredibly subtle in the early embryo, the older tools just couldn't connect the branches. But when the researchers fed the same data into CONCORD, it flawlessly integrated the datasets, filling in all those gaps.
And it went beyond just one species of worm, didn't it?
It did. They threw in data from an entirely different related worm species called C. briggsei. CONCORD aligned the maps across both species seamlessly, and within this complex cross-species space, it accurately resolved what we call fate bifurcations.
Fate bifurcations, meaning like the exact moment a parent cell decides to split into two entirely different identities.
Exactly. For example, CONCORD mapped the precise splitting of specific sensory neurons, the ASE, ASJ, and AUA neurons.
What's special about those?
Well, take ASE neurons, for instance. They developed into left and right versions that sense entirely different chemicals. CONCORD captured the exact continuous trajectory of how their shared parent cell diverges into those distinct left and right fates without breaking the map into artificial islands.
Wow. I mean, mapping a microscopic worm is a massive achievement, but what happens when you point CONCORD at us? At mammalian biology? Because mammals aren't neat and invariant. Our cells are chaotic. They don't just differentiate from one type to another. They're often multiplying and changing identities at the exact same time.
Yeah, this was their second major stress test, mammalian intestinal development. This is notoriously a nightmare for computational biologists because you basically have two competing geometries happening at once.
The loops in the trees.
Exactly. You have the circular loop of the cycle cells proliferating and dividing, and simultaneously you have the branching tree of differentiation cells deciding to become specialized gut tissue.
It's kind of like trying to map the forward movement of a bicycle while simultaneously mapping the circular spinning of its tires.
That's a great way to visualize it. And older tools simply can't handle both motions. They usually force researchers to mathematically regress out, literally delete the cell cycle data just to make the branching differentiation tree visible.
Wait, you have to throw away a huge chunk of biological reality just to make the math work.
Sadly, yes. But CONCORD's bouncer doesn't require you to delete anything.
Because the sampling is so highly resolved.
Right. CONCORD successfully captured both geometries within a single integrated space. You can physically look at the latent space map and see the proliferative cells looping in a circle, while the differentiating cells branch outward from that loop, almost like a spiral staircase. Researchers can finally study how division and development interact, rather than studying them in isolation.
That is profound. And what really blew my mind reading the paper is that CONCORD isn't locked into just reading RNA. It's domain agnostic.
It works across entirely different biological languages. They tested it on ATAC-seq data.
Break that down for us.
So if RNA is a list of active genes, ATAC-seq looks at the physical structure of the DNA itself. DNA is tightly spooled around proteins. If a section is spooled shut, the gene is inaccessible. If it's open, it's accessible. ATAC-seq maps those open and closed regions.
And CONCORD mapped that structural data.
Beautifully.
It even worked on spatial imaging, right?
Yes, using a technology called xenium, which maps the physical X and Y coordinates of cells within actual tissue. They used CONCORD to map the complex physical layout of the tumor microenvironment in human breast cancer.
So it maps worms, it maps guts, it maps the physical space of tumors. What about scale?
Yeah.
Because biology is big.
They ran it on the Tabula sapiens dataset. This is a massive atlas containing over 1 million human cells across multiple organs. When other state-of-the-art tools attempt to process 1 million cells, they often crash. They demand massive amounts of RAM and VRAM.
Because they're stacking those endless hidden layers.
Exactly. But because CONCORD relies on intelligent sampling and just a single hidden layer, it processed the entire million-cell atlas rapidly. It used a fraction of the memory, running efficiently on standard accessible hardware.
Okay, another quick recap for you listening. From perfectly mapping a worm's nervous system to handling a million-cell human atlas, CONCORD's simple sampling strategy absolutely crushes massive computing brute force. Let's bring this all the way home. What does this deep dive mean for you, the listener, right now?
We live in an era that is completely obsessed with the ideology of scaling up.
Oh, absolutely. We are constantly sold the idea that bigger is inherently better. More parameters, bigger data centers, more raw compute. CONCORD fundamentally rejects that premise. It proves that the deliberate curation and quality of the information you feed a system is infinitely more powerful than the raw brute force size of the machine processing it.
And that leads to a really fascinating implication. Right now, Concord relies on brilliant human researchers to design these sampling rules, to program the bouncer at the door.
Right.
But as AI models continue to evolve, the next frontier isn't just human-curated diets. What happens when AI systems become capable of analyzing their own chaotic environments and dynamically designing their own sampling strategies? We might be approaching an era where algorithms don't just process reality, but actively learn how to filter the noise of reality without our help.
An AI that writes its own rules for what data matters. That is a wild thought to leave on. So for you listening, ask yourself, if a simple, single hidden layer network can map the intricate blueprint of life just by changing its sampling diet, where else in our lives, or our algorithms, are we confusing the noise for the actual signal? What complex problems in your own work could you solve, not by thinking harder, but simply by changing how you sample your information? Thanks for exploring the landscape with us. We'll catch you on the next deep dive.